Welcome back to Particle Pharma. In the previous session, I have discussed about cell division, its importance, along with its stages. I recommend you to note down the points which I am explaining as it will be helpful to you and you need not to revise it again and again. It will save your time. It is divided into two phases, interphase and M phase. Again, interphase is divided into three subphases, gap 1, synthesis and gap 2. After gap 2, cell will enter into M phase, which is again divided as prophase, metaphase, anaphase and telophase, which is followed by cytokinesis. M phase is of two types, mitosis and meiosis, which I have already discussed. Based upon the cell type, cell will go or cell will follow mitosis or meiosis. Let me begin with interphase, which is an active phase or crucial step in cell cycle. Before the cell enter into M phase, it will pass through interphase. So it can prepare for division, means cell will get prepared for to divide and to enter the M phase. Next, it will look after the cell growth and function along with regulation and checkpoints. What these checkpoints will do, they will check whether the cell is proper to divide and cell is getting all the nutrients for division, and there should not be any improper cell division. Otherwise, it leads to the formation of cancer cell or tumor. How interphase is divided? In three subphases. What are those subphases? G1, S and G2. We will look into these phases. G1, synthesis and gap 2. G1 phase or the first phase of interphase. It is the longest phase of interphase, I can say. It helps in the cell growth continuously, and it copies the organelles and makes molecular building blocks required for later steps such as these steps or mitosis. Next is synthesis or S phase. It helps in DNA replication or it forms the complete copy of the DNA. Whereas it helps in duplicating microtubule organizing structure which is required for DNA separation during M phase. Next is gap 2. Here spindles are synthesized which helps in binding or keeping the chromosomes on their place. And here also cell growth is observed as we have seen in gap 1, which is required for protein synthesis, and it begins to recognize its content. It will take 10 to 20 hour cell cycles in rapidly proliferating adult cells but can be arrested in G0 phase. What is this G0 phase? This is a dominance phase or resting phase. What it is actually is, I'll tell you. So gap 1 will be followed by synthesis. After that gap 2 will take place. Next M phase will be happening. So this G0, it is a branch of gap 1, or in short I can say it is a checkpoint of cell division. G0 phase or resting phase or quiescent stage or dominance phase. Here the cell, it is neither dividing or it is not preparing for the cell division, but it is in a resting stage by doing its normal functioning. Cell can enter either G0 either permanently or temporarily. Permanently for nerve or muscle cells or aging cells, whereas temporarily in liver cells. Why it will enter temporarily? Because of the lack of sources. As soon as the cell will get resources, it will enter into the cell division cycle. And it is required for cell growth control. Otherwise, it will lead to the abnormal cell division. And it helps in conserving energy when the organism does not require energy, I mean cell division, or when the organism does not require new cells, what it will do is it will stop the new cell division or other cell division, and it will give that energy where it is required. So by this I have completed the interphase and the stage, I mean phases of the cell division. In the next coming session, I'll be explaining about the M phase. So till then, stay tuned, try to get in contact with us, and you can see the cell and mitosis videos on our Instagram handle. In our highlights, you can easily check out. Thank you.